I'm Linda Griffith. I'm a professor of biological engineering and mechanical engineering at MIT, and I direct the Center for Gynepathology Research at MIT, joint with Newton Wellesley Hospital. My PhD research in chemical engineering at Berkeley was focused on designing reactors for large-scale mammalian cell cultures. It was right around the era that therapeutic proteins were being developed for commercial use. I came to MIT as a postdoc, and a next wave of kind of therapies is cell therapies, and got involved in a tissue engineering project right at the very beginning of the field of tissue engineering. As I progressed into a faculty position as an assistant professor, it was an era where there was tremendous phenomenological work in the field, where you put cells on polymers and could see things that looked like or captured features of tissue growing in vivo. One project in particular that I became part of as an assistant professor was to try to grow cartilage in the shape of a human ear for use in plastic surgery. And working with a plastic surgeon at Children's and an anesthesiologist at MGH, we devised a way to make a biodegradable polymer, very simple polymer, the same kind that you use to sew up wisdom teeth when they're taken out. We devised a way to make that in the shape of a human ear so that it would degrade very slowly, allowing cartilage tissue to form when a construct containing cells and polymer was put beneath the skin. That became an icon of the field of tissue engineering and attracted numerous students and other researchers into the field. But what was really challenging at that time is that was a phenomenology kind of experiment. So cartilage cells can kind of do their own thing. The challenge there was making a polymer in a certain shape. What was really missing, though, and what we have in all other areas of engineering, are design principles for how you think about what controls cell behavior, the molecular cues and how they work together. How do you control the cells that are much more delicate, cells like liver cells or heart cells? They're much more fragile, and in general, putting them on a polymer and expecting them just to do their thing in vivo hasn't really worked. So that really launched a whole era in my lab where we started focusing on molecular cues to control cell behavior, and how do we really think about integrating these cues into biomaterials and devices that could be used in regenerative medicine. Those themes are still very important in my lab, and what we have learned over the years is that, it's not surprisingly, biology is very complicated, but happily, there are engineering principles that help you understand how cells quantitatively integrate those cues in their environment. So much of what we've done is not only to make materials, but to interface with numerous other colleagues in our new department of biological engineering, and think about how we use mathematics to describe the way that cells respond to their environment. So in fact, as part of my career, I helped launch this whole new department at MIT, biological engineering, in which we study the way the biological systems work, from molecules to cells to whole tissues and interacting cell systems, so that we can have design principles for building regenerative medicine therapies or other things, ocean ecology, how do microbes work in the gut. All of these things involve molecular, cellular to organismal level of biological interaction. So there was an era of really trying to push development of design principles. There, using materials that control receptor media, how cells adhere, how growth factor receptors get activated, it turns out those were very useful to biologists, and we began collaborating extensively with a number of biologists to both use these as tools for biology, but also to extend them into regenerative medicine applications. And this is still a strong emphasis in my lab with respect to regeneration of bone, liver, and other tissues and organs. One of the most enjoyable aspects about being a faculty member at MIT is the very strong spirit of collaboration that MIT fosters with industry partners. We've had many sponsored research projects in my laboratory that are set up not as companies giving us money to carry out a focused project in the lab, but set up as true collaborations where we have scientists from my laboratory, students and postdocs, going and visiting for short or long periods of time the company labs to understand the needs for science and technology. We've also hosted visiting scientists from companies and structured sponsored research projects so that part of the project is actually conducted at the company. For example, in analyzing drug toxicology, it might be that we provide a bioreactor for use on site at the company location with support for how it works, so that scientists at the company can learn how to do the assays and importantly carry out the kind of complex mass spectrometry or other kinds of analysis that they want on all the metabolites of the compounds. We've also had similar arrangements with development of scaffolds for tissue engineering and other endeavors. The very strong commitment to these kinds of collaborative relationships that MIT fosters is a reason that I find the MIT environment to be one where I really enjoy spending my career.